Welcome. I'm Dr. Émilie Rodenburger, Director of Global Clinical Affairs at Clinuvel. Clinuvel recently announced the completion of recruitment for the company's first phase 3 study of our drug Scenesse in the depigmentation disorder vitiligo. Today, I want to share some insights from our vitiligo program and how we're using key learnings from this study, known as CUV105, to build a dossier for regulatory submission. Scenesse is a controlled-release injectable implant formulation of the active pharmaceutical compound afamelanotide. The drug is approved by the European Medicines Agency, EMA, and the US Food and Drug Administration, FDA, for the rare light intolerance disorder erythropoietic protoporphyria, EPP, where it has been shown to reduce debilitating phototoxic reactions. Our vitiligo program is evaluating the same approved Scenesse formulation as a systemic repigmentation agent, assessing whether it can safely and effectively restore pigment in the skin. To give some context to our program, it's important to first understand the scientific basis for using afamelanotide in vitiligo. With over nearly 3 decades of clinical use, afamelanotide has been proven to induce melanogenesis, that is, to activate the dark pigment eumelanin in skin. Afamelanotide does so by binding to a receptor on melanocytes, the pigment-producing cells in the skin. In short, if a melanocyte is present and well-functioning in the skin, the drug is able to activate pigment production. Vitiligo is believed to be an autoimmune condition where the body attacks and damages melanocytes, often destroying these cells completely. This leads to the loss of pigment in the skin. We don't fully understand why this happens and why it happens on specific body sites, but there is now an in-depth understanding of mechanism by which we can restore functioning melanocytes to these depigmented vitiligo lesions using phototherapy, specifically narrowband ultraviolet B, UVB. I won't discuss this process in detail today, but it is sufficient to know that repeated narrowband UVB phototherapy can differentiate stem cells in hair follicles, which can then become fully functioning melanocytes. What is frustrating for patients and physicians is that this process takes time, at least 3 to 6 months before the first sign of pigmentation can be seen. Afamelanotide with narrowband UVB adjunct has shown the greatest promise in vitiligo. Once there is a melanocyte with functioning receptors activated by narrowband UVB, the drug can bind and exert its effects. The CUV105 protocol is the largest study we have conducted to date in vitiligo patients and compares afamelanotide with narrowband UVB adjunct against narrowband UVB as monotherapy. We completed recruitment for the CUV105 study in May with 210 patients enrolled, 5% more than our target. Ultimately, we involved 37 study sites in the USA, Europe, and Africa, with slightly more than half the patients enrolled at US sites. Our hypothesis in this study is that afamelanotide with narrowband UVB adjunct will result in faster, more widespread repigmentation compared to the monotherapy, with the primary endpoint assessed 20 weeks after the start of treatment. During this treatment window, all patients receive up to 40 narrowband UVB treatments, with half randomized to receive up to 7 afamelanotide implants, 1 every 3 weeks. The scientific and clinical approach to evaluating repigmentation in vitiligo has evolved over time. One tool has been accepted as a primary clinical endpoint to support the registration of a pharmaceutical treatment for the disorder, the Vitiligo Area Scoring Index, or VASI. There are variations of this tool, but it essentially seeks to quantify the extent and depth of depigmentation on a given area and provide a single score where 100 is total loss of pigment on the entire body and 0 is full body pigmentation, no vitiligo. In clinical use, physicians look to reduce the VASI score over time in order to evaluate the impact of an intervention on skin repigmentation. VASI is the tool we are using in our clinical program to assess repigmentation as a primary endpoint, with a focus on T-VASI, or total body repigmentation. Given the systemic nature of afamelanotide treatment and the clinical responses seen in previous studies, the total body evaluation seems to be the most appropriate tool. Secondary endpoints also use the tool to assess the impact of treatment on the face, the so-called F-VASI, given the importance of repigmenting this area to many patients, as well as patient-reported outcomes for quality of life. There are many key learnings from this advanced study of senescence in vitiligo. In the early days of the CUV105 study, we saw a high dropout rate from the patients in the narrowband UVB monotherapy group. We learned that patients who were assigned to monotherapy treatment were not motivated to complete the study. The visible effects of afamelanotide meant that it was obvious for these patients that they were not receiving the implant treatment. To remediate this, we amended the study protocol to include a 20-week extension in which patients on the narrowband UVB monotherapy group could receive the drug with narrowband UVB adjunct after they complete the trial. This revision helped improve patient retention. We expect the last recruited patient to complete their treatment in the fourth quarter of 2025 and first result from the CUV105 study to be ready in the second half of 2026. Our team is currently working on developing the protocol for our next study, CUV107. Discussions with regulatory agencies are ongoing, and the outcome from these discussions, which is expected in the second half of 2025, will inform how we proceed with the CUV107 study. We've made significant progress since Clinuvel announced the start of its vitiligo program in 2010. I would like to take a moment to acknowledge the efforts of the physicians and researchers who are continuously helping to advance research in this field, and especially in reframing the narrative and misconception of this disease. CUV105 is the largest study that Clinuvel has undertaken to date, looking to recruit at least 200 patients across multiple sites around the world. It was a challenging feat, and I'm proud and appreciative of all efforts to date from our site investigators and their teams, as well as our teams at Clinuvel to achieve our recruitment target, and from the ongoing commitment to our study. Finally, I wish to thank the vitiligo community for their continuous support of our research effort. Thank you for listening. We look forward to providing you with more insights in the future.